Everybody. So this is going to be round two. So this is the second video of two. This one is going to focus on meiosis, which is production of the gametes, the sex cells. So that's your sperm and your egg. And as I mentioned in the first video, this is what determines your actual DNA. This is why you are similar yet different from the rest of your family, your parents, your siblings, etc. So I mentioned in the first video that interphase is the same regardless of whether you're doing mitosis or meiosis. Interphase, you have three different parts. You have G1, which is growth 1 or gap 1, when the cell is reforming everything it lost during mitosis or meiosis. And then during S phase, which is the middle part of interphase, that is DNA replication, when you have the DNA synthesis and you double up all your chromosomes. And then you have G2, which I don't think I mentioned in the first video, and that's gap 2 or growth 2. And that's when you start replicating any proteins and structures like the centrioles and animal cells that are going to help you actually do cell division. And that's when you're preparing to actually divide the cell but haven't yet started it. So you're just getting everything ready for it. And so what I'm going to do is I'm going to go ahead and angle this back down the same way I did before and walk you through all the different steps of meiosis and compare and contrast with mitosis. And it's going to be a lot of similar sounding phases and structures and everything, but there's a couple key differences. So we have our normal functioning cell. We go through S phase in interphase. So that's the DNA synthesis, DNA replication. Everything's exactly the same. I have my sister chromatids, which are your DNA after you've replicated but haven't yet divided them. They still exist as chromatin. Everything is exactly the same in interphase. Nothing has changed yet. But the big difference comes in prophase 1. And the reason that's numbered, anytime you have a number after prophase 1, prophase 2, that tells you it has to be meiosis. Because where mitosis, which is the production of your somatic cells, your body cells, only goes through the 1 division, then you end up with the 2. Meiosis, ultimately you do 2 divisions and end up with 4 total cells. And because you have 2 divisions, you have to start numbering them 1 or 2. So anytime you have a number following it, you know you're talking about meiosis. That is guaranteed. And so in this case, we're going to go into prophase 1. And in prophase 1, rather than just having them condense and the nucleus dissolves and they start to get ready to divide, that still happens. But also, and we're going to talk about this more in class and how it relates to Tetris, your chromosomes, so this is chromosome 1 from dad, chromosome 1 from mom, they are now replicated, attached to the hip. They're going to form what's called a tetrad. And so they're going to form a set of 4 now. And then same thing with chromosome 2 all the way down. So they find their counterpart, their pair, and then they bind to it. When they bind to it, they're going to start to do something called crossing over, which is exactly what it sounds like. They start to cross over each other. As they cross over, they can actually swap some DNA. So now what you're doing is you're taking DNA that you inherited from your mom's family and swapping some pieces of it with the DNA you inherited from your dad's family. And so now you are creating a set of DNA from those two families that is completely genetically unique. It has never existed before. It will never exist again because of the way that it's all being set up. And every single time that your body produced more gametes, more sperm or more eggs, it made a unique combination. So no 2 times was ever the same either because it's just completely random. So even like two sperm or 2 eggs from the same person are completely genetically unique. They are not replicated. So now I have my tetrad. I've done crossing over. So that's all prophase 1. That's kind of where the magic happens. That's what makes you genetically unique. Everything else is going to be pretty much the same from here on out. So now I'm going to do metaphase 1, where they line up in the middle, and then they get pulled apart. So that's anaphase 1. And then you end up with telophase 1, where you have the 2 nuclei form, and then cytokinesis. But now there's not going to be an interphase again. There's no second interphase. So you did interphase, prophase 1, metaphase 1, anaphase 1, telophase, cytokinesis, immediately into prophase 2. So you're not replicating again. And so what will happen is now they start to split again. So you can see that these are all splitting. And so then you have the prophase 1, the metaphase 1 where they line up in the middle, anaphase 1 they get pulled, 2. So prophase 2, metaphase 2, anaphase 2, telophase 2. So again, you did all the 1s, cytokinesis, now all the 2s, and then cytokinesis between all 4 of these cells again. And now if you look, I'm going to pull this back a little bit so you can actually see these. You have half the number of chromosomes that you started with, so they're called haploid cells. They have half the number, and they're all unique. There's no combination that's exactly the same between those 4. So you just created 4 gametes, 4 sex cells that are incredibly unique genetically.